This video details our approach to breast cancer prognosis and treatment using fractal dimensional analysis and PMMA nanoparticles. For our prognosis, we will use a personalized MRA tumor detection interface that will be developed as shown here. The first step is to obtain an MRA scan of the patient's breast that shows the blood vessels. Next, we overlay a grid on the MRA scan to create an organized mapping system so that locations determined by the interface can be easily identified on the patient's breast. The third step is to apply fractal dimensional analysis to each grid cell to get a quantitative measure of blood vessel complexity, which directly varies with the extent of angiogenesis. We then get a single number called the fractal dimension, which is what you see in each of the grid cells. We will then extract the at-risk grid cells. The higher the fractal dimension, the more chance there is a tumor in that cell location. Looking at the MRA scan, we can easily see that cell C5 has the highest fractal dimension of 22. Our interface will output a table of the areas that are at the highest risk of containing a tumor. The next step is to create a 3D visualization that quantifies the tumor and makes it easier to see which areas are at the highest risk. For treatment, we will use a thermal injection of PMMA nanoparticles directly into the site determined by the interface. At the cellular level, each PMMA nanoparticle binds to a receptor on the surface of a tumor cell. It then releases the encapsulated drug molecules into the cell. Using the thermal energy of the exothermic polymerization process, these molecules will inhibit the signaling cascade of TGF-beta and ANGPTL4, two signaling molecules implicated in tumor metastasis and growth. This means that the tumor cells are reduced in size and thus eliminated. Thank you for watching this video.